much. The first speaker is Professor Jean-François Arnal. He is an MD PhD. He's a professor of medical physiology at the Paul Sabatier University in Toulouse. He is also a senior scientist at the Institut des maladies métaboliques et cardiovasculaires of INSERM. He is also a senior member of the University Institute of France and also, obviously, a foreign member of our company. He has been president of the French Society of Atherosclerosis, and he has a team that is very well known worldwide for the last 25 years in the basic knowledge of physiology and medicine of estrogens and their receptors, and made major contributions in the understanding of these treatments and also the new classes of SERMs, the selective regulators of estrogen receptors and progesterone receptors. So, Professor Arnal, thank you very much for being with us today.
Thank you, Mr. President. Dear colleague.
So it's really a pleasure and an honor today to participate to this celebration in the honor of Jean-Michel Foidart. So I will try to select what can explain, in fact, the mechanism of action of estrogen at the level of the molecular biology. So here is the structure of the main estrogens, and the estradiol has here a planar structure, hydrophobic structure with two hydroxyl groups, and estriol and estetrol has 3 and 4 hydroxyl group in their structure. And what you know is that estrogen receptor is a nuclear receptor and transcription factor. And this is the classic scheme with the estrogen receptor here, its translocation to the nucleus, and most of the action of estrogen receptor until recently were known as transcription factor here, after binding the DNA here, and eliciting gene expression. The estrogen has pleiotropic effects in women that can be modelized in mouse. And the beneficial one in women, for instance, the prevention of coronary events before 60 years, prevention of diabetes type 2 or osteoporosis, can be mimicked here in mouse models, as well as the proliferative actions of estrogens at the level of the breast or of the endometrium that can be deleterious as they can promote cancer, can be opposed in the endometrium with the progestin. And finally, the action of estrogen at the level of the liver through an induction of coagulation factor can promote, can increase the risk of venous thrombosis. And thanks to this model, we have tried to understand the mechanism of action of estrogen in vivo, mainly in mouse models, and we focused on the vascular aspect, on arterial aspects, that are mainly protective. And with Francis Bayard, 20 years ago, we began to modelize this in mouse, and we found a huge protective effect of estrogen on atheroma, a prevention of the neoepithelial aplasia, is to say a prevention of the restenosis of an artery after injury, and surprisingly, we also found a very important role of estradiol in altering flow-mediating remodeling, it is to say an increase in the size of the arterioles in response to an increase of flow to match with the tissue demand. We began a fruitful collaboration with Pierre Chambon in Strasbourg. And at that time, one question was, because there are 2 estrogen receptors, ERβ and ERβ, what is the most important one? Because there was a controversy at that time due to a leakage, in fact, of the first mouse model of inactivation of ERβ, that was done by Ken Korak, and because of the leakage of these mouse, the inactivation was incomplete, and so there was still a chimera of receptor that was still produced and that accounted for some actions of residual action of estrogens. And thanks to a second generation model generated in Strasbourg, we were able to demonstrate, there's the picture of Pierre Chambon here, sorry, And thanks to this model, we were able to demonstrate that ER-alpha, in fact, mediates most of the action of estrogen in vivo. So we focused on ER-alpha, and we tried to understand how it works. In fact, ER-alpha here is structured with a DNA binding domain, which binds to the DNA, ligand binding domain, which binds the estrogen here, and with the conformation that's line the binding induces to hold the molecule. And these changes in conformation will activate here AF1 and AF2, that are the two main activation functions of the molecule. Here you have the timer, which once activated after binding estradiol, will recruit here cofactors, and in fine will recruit the RNA polymerase and will induce numerous gene expression and in fine the biological action of estrogens. So we continue this work with Chambon, and we inactivated respectively and selectively AF1 and AF2, the two activation functions, and we found that whereas AF1 has just a synthesizing role of the two estrogen, AF2 is absolutely required, and if you kill, by mutating several amino acids here, if you kill the AF2 function, you kill most of the action, all the action of estrogens, and the phenotype of this mouse is quite similar to the full knockout of ER-alpha. So at this step, we found that for all these actions, as well as numerous other actions, at the level of bone, at the level of the uterus, you have no longer any action of estradiol. And in fact, one key was given by this fourth model of pathophysiological artery with carotid, with a model of carotid artery injury. It consists in destroying both endothelial and smooth muscle on a precise length of 4 mm, and then you treat the mouse with estradiol here. That accelerates the reendothelialization compared to an animal that is not given estradiol after castration. And you can quantify this process. You can see here acceleration of reendothelialization by estradiol in wild-type mice. This effect is completely abolished, as I mentioned before, in ER-alpha deficient mice, in mice without ER-alpha. Normal action of estradiol in mice with ER-beta. And very surprisingly, in mice where we have killed the AF2 function, the nuclear action of estrogen, we still have a response to estradiol. And this is the beginning of one new aspect of estrogen. Because if you kill here the action of the receptor in the nucleus, it remains, in fact, subpopulation of estrogen receptor outside the nucleus. And this was studied 10 years ago by John Katzenbogen, thanks to a very original molecule that is estrogen dendrimer conjugate. It is a macromolecule with numerous estrogen bound to this macromolecule, and this macromolecule can enter the cell and stimulate the membrane receptor, but is too large to enter the nucleus. And thanks to this molecule, he was able to demonstrate that activation of this membrane receptor here is extranuclear receptor is able to accelerate reendothelialization. Then we raise the question, is membrane estrogen receptor necessary for this effect? And to answer this question, we mutated, in fact, here in the estrogen receptor, one amino acid that is responsible for palmitoylation. Palmitoylation is a lipidation, a modification of the estrogen receptor that allows it to be targeted to the plasma membrane and to remain so outside the nucleus. And we found that after killing this membrane receptor, we abrogated the action of estradiol on reendothelialization, whereas the uterine growth is completely normal. So here, estrogen receptor at the membrane is dispensable for uterine action. And conversely, you have here again the mouse AF20, where the nuclear action are killed, and you still have an acceleration of reendothelialization thanks to the remaining receptor at the membrane here, but the uterine remain atrophied. So let's now move to the estetrol, and how can we explain the action of estetrol thanks to this molecular background? So Jean-Michel Foidart already mentioned this aspect of estradiol, and one very important point is that estetrol, that is very abundant here in the human plasma fetus, its role is completely unknown from a physiological point of view. So we try to understand what is the effect of estrogen estradiol and estetrol after binding to estrogen receptor through crystal structure binding approaches or through binding approaches. And when you study the conformation of the receptor after binding of these two estrogens, you cannot, in fact, really find differences. So when you isolate the nuclear estrogen receptor, there is no real difference. Similarly, when you study the effect on uterus, we previously found, and that transgenic mouse and pharmacological model demonstrated that the uterine growth, the endometrial proliferation, relies entirely on the nuclear ERα action. And accordingly, estetrol here has the same effect on gene expression at the level of the uterus than estradiol. So again, the effect of estradiol and estetrol on uterus are very similar. And one major difference was given by the fourth model of vascular injury. It is to say the model allowing to study reendothelialization. And again, you see that estradiol is able, as estriol, is able to accelerate reendothelialization through the stimulation of the membrane receptor, but not estetrol. And if you add simultaneously estradiol and estetrol, you even block the reendothelialization by estetrol. And thus, estetrol differs profoundly from estradiol because it does not activate the membrane receptor, but it is also able to block the action of estradiol on the membrane receptor. And so, first, to summarize these different aspects, we can say that, and I hope I have convinced you, that estradiol is able to activate both membrane and nuclear receptor, whereas estetrol acts only on nuclear receptor and is not able to activate the membrane receptor, but is even able to block the estrogen action on the membrane receptor. And the counterpart of that in vivo is that some tissue rely mainly on the nuclear action, for instance the uterus. Some endothelial action as reendothelialization, endothelial healing, rely mainly on membrane receptor, and other tissue, as the liver, is a mixed tissue that depends both on nuclear and on membrane action. And this is the first level of explanation of what we will see later. It is to say the little effect of estetrol at the level of the liver. And how can molecule bind differently from the same product of a gene? It's probably here the palmitoylation and the numerous molecular interaction of this receptor that allow this entity to be recognized differently by estetrol and estradiol. Is it so simple? Not really, in fact. And to conclude, these are very unpublished data here, where you can see, thanks to single gene single-cell gene expression here, single-cell RNA-seq, that allow you to characterize, in fact, gene expression in the cells of the aortic vessel. And you can see that you can characterize, so this different population of endothelial cells, smooth muscle cells, fibroblasts, and macrophages. And you can see here that estradiol and estetrol induces gene expression changes very differently here in this subpopulation. A lot of changes here in the macrophage in response to estradiol, for instance, and a lot of changes in smooth muscle cell or fibroblasts in response to estetrol. And so now we have to investigate further why these gene expression changes are so different. And the epilogue of this, of that is that in a different model of injury, it is to say now, in a model where you kill only the endothelial cell and not the smooth muscle cells here, which gene expression is differently changed by estetrol. In that case, estradiol and estetrol both accelerate reendothelialization, and this acceleration of reendothelialization in that case by estetrol is not the consequence of an effect on the membrane receptor, but a direct effect on the at the level of the ER-alpha nuclear receptor in smooth muscle cells, because if you kill selectively the ER-alpha in the smooth muscle cell, you abrogate the action of estetrol. So, to conclude, estradiol elicits both nuclear and membrane action, whereas estetrol only activates nuclear actions and not the membrane action. And depending on the tissue and cells, you can have similar action at the level of the uterus, a lesser action, lesser hepatic effect by estetrol, and in arteries, it's as most of the action on nuclear, you have the similar protection by estetrol than by estradiol, and with some time, some mechanism that remains to be elucidated. So, estetrol appears as a singular natural estrogen whose physiological action remains to be discovered, but now the medical use will be detailed, and I would like to thank all the people, in fact, that contributed to this work, and in particular, I will now switch to French for 1 or 2 minutes je voudrais terminer par, si on reste un peu plus personnel, pour Jean-Michel Foidart. Donc, un des bonheurs que m'a réservé ce parcours de médecin-chercheur a été la rencontre de personnes remarquables, inspirantes, avec qui j'ai eu le privilège de travailler, et vous en avez vu un certain nombre ici. Ainsi, il y a 13 ans, j'ai rencontré Jean-Michel Foidart, grâce à un congrès organisé par Andreas Gennadiani à Pise. J'ai aussitôt été attiré par sa prodigieuse connaissance de la gynécologie obstétrique, mais aussi par son extraordinaire curiosité concernant les aspects moléculaires. Et ces modèles et ces outils que nous avons développés à Toulouse nous ont permis de répondre, enfin je l'espère, au moins en partie, à un certain nombre de mécanismes de l'estétrol, et ainsi à répondre à certaines de ces attentes. Mais bien des mystères demeurent, au premier rang desquels le rôle physiologique de l'estétrol pendant la grossesse. Il y a donc encore du travail pour nos successeurs. Durant toutes ces années, j'ai pu apprécier ses multiples qualités, à la fois remarquable médecin, très grand chercheur, fantastique organisateur et véritable homme orchestre dont témoignent, entre autres, ses actions dans cette académie. Cher Jean-Michel, je tenais donc à te remercier chaleureusement, dans notre langue commune, pour tout ce que tu nous as apporté, tant sur le plan professionnel que personnel.
We learned a lot, and we all have a lot of questions. So the way we will manage this is first we will ask questions from the audience here that is physically present, and then Madame Costanzo will manage the questions from Academy members that are on Zoom and public. So questions from the audience. Yes, Patrice Cagney.
Thank you for this very nice lecture, very interesting. I have a very basic question. I'm not familiar with this, this topic. I was wondering if the palmitoriation of the receptor, the membrane space, if we know how it's regulated first, and if some nutritional conditions can change the activity and finally can contribute to explain some of the relationship between obesity, type 2 diabetes, and this ER alpha at the membrane site. Thank you.
Thank you for this question. It's obviously a very good question, a very important question. In fact, the first point is that the membrane receptor is not abundant at all. It's very difficult to isolate this receptor, and even it's very difficult to see this receptor. We have tried by many ways, by adding a tag on this receptor through antibodies, and we have a lot of trouble just to see this receptor, and so it's not easy to characterize its abundance. So we have indirect evidence of this receptor, many evidences, but the direct visualization is still very difficult, I would say, not really reliable so far. And so the palmitorylation is a reversible modification, and this palmitorylation, the regulation of this palmitorylation is not really. Palmitorylation concerns many other molecules, so we don't have the possibility to really investigate the action of changing the abundance of palmitate, for instance, or the enzyme that lead to this cross-translational modification. So it's an open question.
Thank you very much,
Yes, thank you for this fascinating presentation. My question is, do you have the same side effect with Estetrol given in male animals than other estrogen in male animals?
So the male issue is an important one, because you know that in fact males have a receptor almost as abundant as in females, and we don't have studied extensively the action on males. We have studied mainly females, but as for other estrogens, it seems that you could have most of the action in males than in females. Sometimes you're obliged to double the dose, because males are a bit less sensitive for probably epigenetic modification. And so we can expect to have, I would say, rather similar action.
When we teach in cell biology this chapter, we talk about some chaperones that bind these receptors. Do you have any evidence whether the chaperones that bind the membrane one versus the cytosolic moving to the nucleus one are different?
Again, it's like for the palmitorylation. It's very difficult to isolate these molecular complexes because they are very labile, and so it's all this interaction appears very transient. So it has been rather easy and more easy now to study what's happened at the level of the nucleus, and we have many tools for that. Outside the nucleus, it's it's much more difficult, because it's probably submitted to phosphorylations, and these effects are very transient. So it's very difficult to have a picture with time.
But the model eventually is that even this membrane receptor with other partners ends up also regulating genes, right? So on the chromatin, you should have a different complex.
So there have been many studies, mainly in vitro, trying to see what are the interaction between membrane and nuclear action. And so obviously there is some impact of the membrane action to the nuclear action. One additional possibility that is nicely worked by a colleague in Rennes, in France, is that this action could occur at the level of the RNA. And so the translation could also be influenced by estrogen. So I would like to conclude with the fact that the estrogen receptor appeared very early in evolution. It was the first, the father, in fact, of all the steroid receptors. And it's probably the reason why it's so nicely integrated in our physiology.
Thank you very much. Well, it's time also for the outside world to have access.
Yes, but the outside world seems to be very quiet this morning, so I guess